kind of a lot because we covered a lot. Let's see. Got too much stuff open. So going back to the very first what we started talking about. Um, Biology itself is science, right? That means that everything that we look at in science has a supporting evidence, and the way we get this evidence with all sciences is the same way. We use that scientific method, right? So with the scientific method, what should you be doing for the scientific method, right? What's the very first thing that we need for any sort of experiment? Remember? Might be my next one. Yeah, this next one too. Oh, there it is. This is a little easier. Let me share my whole screen. Entire screen. That's the one I want. Yeah, there it is. So with the scientific method, it's pretty much this thing here on the right. You're going to make a hypothesis, and then from there you're going to do a test called an experiment. With the experiment, you collect your data, and then you run analysis, and you want to get that information back out to the public, so you share it. So that's the whole scientific process. No matter where you go, you should be using this method to acquire data. So with the hypothesis, anybody remember what we make or what makes a good hypothesis? So that's going to be something that you it's referred to most of the time as an educated guess, but it's a little more than an educated guess. It needs to be testable, and it also needs to be falsifiable, meaning that if you were to try to do an experiment, you need to be able to prove it wrong, not prove it right. Um, and then again, you need to be able to prove or test it, meaning that it can't be something um, paranormal or supernatural, like with luck. Luck's not something you can test or measure, right? So how would you measure somebody's luck? And then when you go after you formulate a hypothesis, you build up an experiment around it, and then from that experiment, you're going to be looking at different variables in your experiment. So these variables are dependent on what you manipulate, and then what's being manipulated by the other variables. So when you go in and you change variables around, that variable is going to be called my independent variable. And then from there, the thing you manipulate, it's going to affect something in your experiment. So the effect is going to be my dependent variable. So let's say that I'm doing an experiment where I'm looking at plant growth of different shades of light. So I'm going into this setup and I'm changing the colors of light, red, blue, and green. That's going to be my independent variable because I physically change that. And then I'm going to allow the plants to grow. And then after they grow for some time, I record the height. So that growth in height is my dependent variable because that's what was affected by the light. There's also other variables in the setup. They're called constants. So constants are going to be the same throughout your experiment. You don't want to change too many variables because it might be difficult to determine what is causing the change if they have this multiple um, changing around an experiment. So these are things you want to keep the same. So if I was doing that experiment again on plants and colored light, I would want to make sure that all the plants were getting the same amount of water. They were getting the same amount of um, fertilizer. They were in the same location. Like you didn't have one outside and one inside the house. So again, these are things you would want to keep the same. And then you usually have a control with your experiment. Controls are going to be usually what's referred to as uh, the for change. So if I was doing an experiment with plant growth, I would want to have one in natural sunlight, and that would be my control. That way I can use that plant as kind of a default or a base to see how much changed when I change the light. From there, you just run analysis. This is where you do things like means, median, and modes. And then you would want to get this information back out to the public. And that's where you would want to publish that into a book or a journal and have people read it and look at it. And then you also accept or reject your hypothesis from there too. But again, everybody should be doing the same thing. So again, biology science. So specifically, biology is the study of life. And then with biology, how do we define something that's living? Anybody remember a characteristic you have to have for to be a living organism? So these are all my requirements, right? If something fails one of these requirements, then they shouldn't be considered living. So growth, usually growth is coming from the fact that cells get bigger, and then when cells get big enough, they would want to divide. So that would be something like mitosis or fission. Cells need to be able to move around. So again, with animals, very easy to see because we have muscle tissue, and that muscle tissue moves. Not as easy to see with bacteria and other smaller microorganisms, but they still have the ability to pull water in. They still might have flagella or cilia, so they can still move. Plants can move too, even though they're stuck in the ground. They're able to pull water from soil to leaves, so it's still going to classify as movement. They need to be able to reproduce, so this is going to make a either identical clone, which is going to be mitosis, or asexual reproduction, or they need to be able to make a offspring. They need to be able to respond to stimulus. So again, very easily seen with animals. We have the ability to sense light, sense temperature, sense taste and smell. That's all stimulus. Plants not as easy to see, but they can sense light. Bacteria can also sense their surroundings. Metabolism. So if you don't remember what metabolism is, it's the sum of chemical reactions in the body. It could be fast or slow, but it is chemical reactions that organism is doing. And then homeostasis. Organisms need to be able to maintain a balance, and then that balance is going to keep them normalized. And usually, if they don't come back from that balance or they change, that's usually a disease. And most, most things are very easy to see with these requirements. There are some organisms that fail some of these requirements, like viruses. We talked about viruses not being living. That's because viruses, they don't meet the metabolism checkpoint. They have no metabolism. And then for the reproduction, they actually need a host. So without that host, they wouldn't be able to reproduce. So again, they fail those two. So they're not going to be considered living by many people. I think we moved on to cells next. Science. Or, it's chemistry. With chemistry, I think the main takeaway here is water is going to be considered the life molecule. Without water, life really wouldn't have originated. All the aqueous, um, how about this? All the metabolism in your body, they're all aqueous chemical reactions. So without water in your body, you would not be able to perform any sort of metabolism. Let's see. Another really important property that water has is the ability to form hydrogen bonds. So those hydrogen bonds are going to be important because they keep you from overheating. Whenever you get too hot, you don't just like spontaneously burst into flames. That's because all that water in your body is able to absorb heat for you. It's also going to help dissolve ions like salt or sodium chloride, calcium chloride. All that material in your blood is going to be ionized because it's dissolved in water. Let's see. With the biological macromolecules, several of these are very important for your like diet. You have carbohydrates, which are sugars. You have let's see, lipids and proteins. You also have DNA. So all of these are going to be considered biomolecules. because they're found in living organisms. They're also organic molecules. Anybody remember what organic means? How do you distinguish organic versus inorganic? So that is a very easy distinguish. If I can talk, easy distinguish because of carbon. So all these carbons you see in this chain, as long as there's a carbon with the oxygen bond, I mean carbon with the hydrogen bond, those are going to be organic molecules. So something like sodium chloride, NaCl, it lacks carbon, so that's going to be an inorganic molecule. Okay, here's our cells. So cell biology is looking at the smallest unit of life. You can't get smaller than a cell, but then you're not going to be considered living anymore. So cells the smallest unit of life. The two main types of cells in our Earth is going to be a prokaryote versus a eukaryote. So a prokaryote, like this one shown on the screen, it does not have a nucleus. It does have DNA, but the DNA is not encased inside of an envelope. If you look at a eukaryote, like the one on the bottom of the screen, it also has DNA, but that DNA is being trapped inside of this nucleus. So those are the two main types of cells. They're going to have similar structures. So you might see cells with cell walls. So cell walls are these thickenings around the plasma membrane. Cell walls are very good at keeping the cell shape. They also protect the cell from too much water. So most of the cells that live in water need to have a cell wall. Um, our cells, our animal cells, they do not have a cell wall, so they are very prone to swelling and bursting if they have too much water. Um, that some cells can have flagella, so that's a long whiptail-like structure here. And then you have pili. So pili are going to be these bristles that are used to help clean the surfaces. You might see cilia, which are also little hairs, but these move around. Okay, so looking at some of the organelles, we talked about the plasma membrane. So every cell needs to have a membrane that's going to maintain its shape or form its shape. It's maintained by the cell wall. Plasma membranes are made up of phospholipids. Everything on the inside, the liquid part, that's called cytosol. And then the little solid units, those are organelles. And then if you take the liquid plus the solid, you get all of that kind of jelly matrix. That's called cytoplasm. It's all cytoplasm. Other organelles that we talked about, chloroplast. So these are your photosynthetic chloroplasts. So these are only found in things like plants and algae. Animals don't have those. The nucleus again is only found in eukaryotes, and it holds DNA. The mitochondria is found in both plant and animal and fungus. So it's this one here. It's called the powerhouse of the cell. It's not found in bacteria because bacteria and mitochondria are roughly the same size. Lysosomes are found in larger eukaryotes. So a lysosome is a cell that's going to help break down and clean, usually destroy matter that needs to be destroyed. Ribosomes are these little dots here. So these are the protein builders. Without ribosomes, we wouldn't be able to produce any proteins. The area where these red dots are, that's my rough endoplasmic reticulum. So that's going to be used to build up proteins. So we have the Golgi apparatus. Think of this as the post office. This is going to receive proteins, ship proteins, even sort the proteins for you. We have our centrioles, which are this area here. These are going to be producing microtubules for movement of organelles. The cytoskeleton, which is the green here, that's going to maintain the shell shape. It's going to aid in mitosis when we talk about cytokinesis. And then you have vacuoles. Plants have a very big vacuole called the central vacuole. So that's where the area they hold water. So if a plant is wilted, it's because the central vacuole is losing water and the cell's collapsing in on itself. So this is a zoomed in version of the plasma membrane. Again, very important complex here for the cell. It's maintaining the cell shape, is also maintaining what stays on the inside and stays on the outside of the cell. Because the phospholipids you see in red, they are bipolar molecules. You have the polar heads, which are on the outside and also oriented toward the inside, and then you have the non-polar tails in the middle, kind of sandwiched together. So if there's anything that's polar on the outside of the cell, it's pretty much trapped on the outside because it can't cross non-polar. And same thing on the inside. If there's something polar on the inside and tries to get out, you really can't because of the non-polar region is trapping it on the inside as well. So to move things across, you're going to have to use the blue structures. So these blue structures are proteins. There are some like this one here that's a channel. There are some that are pumps. So channels are usually going to be passive, allowing movement across without aid of ATP. But pumps are going to be using ATP because they have to be paid in order to pump the molecule across. This is also referred to as fluid mosaic. So that means that these blue molecules, these proteins, they're not trapped in their specific locations. So during time, as this is a fluid membrane, these blue proteins are able to kind of move around like little ships. So if you were to look at the cell surface one day, take a map of it, and then come back another day and take a map of it, they're going to be completely different because these proteins are able to move around. So with the movement, you can um, name it based on whether or not ATP is being used. So again, passive movement is going to be something that does not use ATP. So passive is free. Types of passive, you have diffusion, facilitated diffusion, osmosis. So these two go together. Diffusion is just high concentration to low concentration. So a good example here. On one side of the membrane, we had a lot of this molecule. Over time, though, it starts to seep down and across the membrane, and now it's kind of equal. So that's diffusion. Once it's equal on both sides, it's not going to want to change concentration anymore. Facilitated diffusion is the same type of movement here, but we're going to be using a protein. Usually there's a channel here in the middle, like a straw, and the proteins are going to be going down that straw. Osmosis is also passive, but osmosis is specialized to water movement only. Um, Usually with animal cells, it's very um, dramatic, the amount of water that can move across the cell sometimes. It's all dependent upon the salt located. So if there is a lot of salt on the outside of a cell, it'll actually pull water out because it'll dehydrate the cell. If you have more salt on the inside of the cell, like if you were to drink pure water, the water would rush into your cells where that salt's at, and it would cause your cells to swell. And then we have active. So if you have active transport, the molecules are going to be pumped across with help, and that help has to be paid ATP. So you are being kind of charged for this work. With the movement, you have phagocytosis, pinocytosis. So phago is going to go back to eating. So we're moving across solid molecules. So like this big particle here is bacteria. It's being trapped in a vacuole and then being pulled in. So that fact that it's being pulled in is going to be using ATP. Same thing over here with the water. We have water and salt being trapped inside of this little vesicle, and then it's going to be pulled in. And when it's pulled in, you had to pay for that with ATP. I'll click. We looked at respiration. So with respiration, it's the process of making ATP. There's two ways to do it: aerobic and anaerobic. Um, it is a transfer of energy. We don't want to break down the molecule that we're using to build ATP very quickly with, because if we were to, we would use a lot of that or lose a lot of that energy as heat. So we actually break it up into several steps. It's called a metabolic pathway. And each time you do a different step of the pathway, you harvest a little bit of energy. So again, you can throw sugar on the table. Again, our glucose is our go-to molecule, and put a flame to it, and the whole sugar will just burn up in flames. But we don't want that happening in our body. We want to break it up into steps. Oops. So the first step, if you're using oxygen or not, is always going to be a glycolysis. So glycolysis is going to take your glucose and it's going to break it up into two molecules. Now these two molecules are called pyruvate or pyruvic acid. And then from here, you do harvest a little bit of energy, but that's not the main thing being harvested here. You do have this thing called NADH being created. The NADH is important for the very last step of this phase because that's where we're going to get the majority of our ATP. So from here, the pyruvate can go into the mitochondria for aerobic respiration, or it can be transferred into lactic acid outside the mitochondria. We'll look at the aerobic one first. So we've moved into the mitochondria right now. We're going to keep going through the pathway. So notice how we have the pyruvate that entered. Again, we're in the mitochondria. And then we have our little steps here. So each step does a little bit of conversion. We had some NADH created right there. We have more NADH created there. There's some FADH, which is another molecule that's transferring energy. And notice how we had carbon dioxide too. So carbon dioxide is the byproduct here. And then whenever you exhale out carbon dioxide, this is where it's coming from. This part of the respiration phase. And then we made a little bit of ATP, but we haven't made much. We haven't made a lot just yet. We've only made about 10 so far. We can get much more with this last step. So this last step is called the ETC, or the electron transport chain. This is found inside the membrane of the mitochondria. And then we take those NADHs and those FADH that we created in the first and second step, we use those here. So we also refer to these as electron taxis because on the NADH and the FADH, we have hydrogen, and then along with hydrogen is a proton and electron. The electron is passed to this protein complex. You can see it happening right here. So NADH hands off the electron. I can make it bigger. Make it a little bigger. And then the electron goes down the blue proteins. That actually produces a little bit of electricity. And then the electrons are handed off to water, or oxygen to make water. And then you notice that didn't have any ATP, right? The ATP is coming from the fact over here with this protein. This protein is called ATP synthase. So all these hydrogens are being pumped across. They want to get back down to this middle. They actually can't because of this membrane. The only way they can is through this one channel. And if they flow down this one channel, again, it's moving a positive charge. So again, it's a little bit of electricity. And that's going to generate a little turbine here. And as it spins, it converts ADP over into ATP. So that movement of hydrogen down that gradient is going to give you roughly 26 ATP from just the molecules you harvested in the first two steps. So overall, you get around 36 to 34 ATP with just one sugar molecule. And that's a really good payout if we use oxygen. If we don't use oxygen, we're going to be doing anaerobic respiration, and that's going to just make lactic acid and 4 ATP. So it's a very poor payout, but the benefit there is very quick. It's a very fast turnaround, so if you need energy very quickly, you have another way to make it. I think, yeah, right there it is. So notice how we have the glycolysis, pyruvate, but from here we don't go into the mitochondria. We stay outside the mitochondria. We can't use oxygen there. And we just turn over and make lactate, which is lactic acid. You get 2 ATP from that reaction plus 2 from the glycolysis, so 4 total. The only benefit for this is it's very fast. You couldn't survive on this in the long term, but it is very beneficial for a short-term use. Other organisms do this too. The fact that you see these beans in the background, those are being used to make alcohol or cheese or something like that. They're being deprived of oxygen and they're fermenting inside of those big barrels or big beans. We moved on to photosynthesis next. So photosynthesis is the reverse of respiration. It's actually the creation of sugar, not the breakdown of sugar. The only organisms that are able to go through photosynthesis need to have some sort of pigment, like a chlorophyll or um, chloroplast, either one. So the difference there is chloroplasts are the organelles, chlorophylls are the pigments. So something like bacteria will probably have chlorophyll, while algae and plants have the bigger organelle chloroplast. Down here is my equation. So we're going to take carbon dioxide and water, which are the byproducts of our respiration, and we're going to build them back up into sugar and oxygen. Again, only photosynthetic organisms can do this. So this is broken down into two steps: the light cycle and the dark cycle. In the light reaction cycle, you're going to be harvesting light. So over here we have our, let's see, yeah, this is what I want. We have our chlorophyll. The chlorophyll is absorbing light. It's getting heated, getting some energy, and then as it heats up, electrons inside the chlorophyll are going to vibrate. Eventually, they're going to vibrate enough to where they jump out of this into a complex. They're going to move down the complex into another chlorophyll pigment. It's getting excited again. It jumps out, moves down more proteins, and then it's going to jump onto NADPH. So the NADPH, we'll come back to that. We go to the next phase with it. But with these electrons, they have to be replaced because if you're losing electrons, it's going to be bad if you keep losing and don't get them back. So you actually split, or plants, they split water. And when they split water, electrons jump off of water. They refill the electrons here. And that's where you're going to get half of your oxygen from. And then another water molecule break and give you a full oxygen molecule. Uh, Same thing here when the NADPH is made. Some proton are being passed across the membrane. They're going to want to go through the same ATP synthase complex back down the gradient. So very similar to the respiration phase. As this goes down, it spins a little turbine, makes some ATP, and the ATP and NADPH, these two are going to be very important for the next step. We're actually going to use those to build up sugars. So with those two molecules the plant's made in the first phase, you're going to add in the carbon dioxide now. So you start out with carbon dioxide, you add in make this bigger. You add in the ATPs, the NADPH is created in the first step, and you take one carbon and you build it up into a three carbon. And then from there, that three carbon molecule is going to be processed and made into sugar. So it's the complete opposite of cellular respiration. The next topic we looked at was cancer. Uh, Cancer is affecting a lot of people and they don't really understand what it is. So, what cancer is at its definition is just the inability for a cell to stop mitosis. It's pretty much stuck in back to back mitosis. When it starts to lose control, the cells don't really have the ability to um, maintain that balance anymore. So they keep producing, keep copying, and they kind of layer on top of each other. And as they layer on top of each other, this structure that starts to form, we refer to it as a tumor. And tumors, depending on their location, have two names: benign or malignant. So a benign tumor is usually small, and they don't really grow into different organs. When they grow and expand, they have yet to grow and break their original barrier. But with malignant tumors get bigger and bigger, or they're very close to a transport system like a blood vessel, and they break out into that new area. And if they break out into other body parts, especially the blood vessels or the lymphatic vessels, we're going to refer to those as malignant. And then those are the bad ones. The ability of them to do that is called metastasis. Just when they grow, they can break the tissue barrier, and breaking that tissue barrier is metastasis. Factors for cancer, there's two main reasons really. You have genetic and um, environmental. So genetic is usually predisposed cancers. They're caused by mutagens. So a mutagen is going to be a gene that is very closely um, about to mutate, or it's not far off from mutating. Um, Good example was BRCA1. So with BRCA1, it's a gene that controls uh, cells in breast tissue that are going to. It was a gene that allows for cells to stop or go through apoptosis if they need to, tumor suppressor gene. But if it mutates, it's kind of like cutting the brakes on a car. That gene is going to stop working, and if it does become cancerous now, it doesn't have the ability to stop mitosis anymore. The other reason for cancer are carcinogens. So with carcinogens, these are going to be factors in the environment that can mutate DNA. A really good example is sunlight. So sunlight is UV radiation. So this UV radiation is extremely high in energy, and it's high enough to pierce our DNA, kind of jumble up the DNA, mix it up, and if you don't put it back together correctly, it could cause a mutation. Other factors like alcohol, cigarette smoke, anything that really could rearrange DNA is going to be considered a carcinogen. So this is going over the cell cycle. This is a normal cell. With the cell cycle, you should have the start of interphase being G1, middle of interphase being S phase, the end of S phase, or S phase, interphase being G2, and then from there you enter the next phase, which is mitosis. So you can see here that interphase is a very big part of the circle, so cells should be in interphase the majority of the time, and mitosis is a very small piece of this segment of the circle, so a very short amount of time. If a cell was to become cancerous, it kind of skips all this big part of the circle, and it's back to back mitosis. There's no regulation of the mitosis when the cell becomes cancerous. So there's all sorts of problems with cancer cells. This is because look at the pattern here. So the first thing after mitosis is going to be growth of the cell, getting back to the original size. So you have abnormal size cells with cancer cells. DNA synthesis. This is where you're going to have the cell copy its DNA, for every daughter cell produced will have the right amount of DNA. So you can look in cancer cells and you'll see that the DNA chromosomes are all messed up out of the correct order or number. And then after that, you have rearrangement of the cell trying to get ready for mitosis, growing microtubules and stuff like that. With mitosis itself, very easy to distinguish the phases. You have these five phases really: prophase, metaphase, anaphase, and telophase. Prometaphase is kind of the middle between pro and meta. So what you should expect with prophase, the cells coming right out of interphase. So it's going to have a lot of characteristics of interphase at the very beginning. So you'll see a nucleus. You won't see chromosomes fully formed. They're all kind of, not all, some are going to be mostly chromatin. You can kind of start seeing the wormy looking structures appear. The nucleus is still present again. The spindles or the centrioles, they are close together. Their microtubules haven't developed a lot yet. But by the time you get to the end of prophase, like here in prometaphase, the nucleus is gone completely. The chromosomes have fully condensed into a solid structure. Spindles are on both sides, are opposite poles of each other, and you should be able to see microtubules formed. So good examples down here with these images caught by a microscope. Now with metaphase, metaphase is easy because the chromosomes get pushed or pulled, and they're always aligned down the equator, the middle of the cell. So right here with alignments is going to be metaphase. Anaphase is going to be where the chromosomes are being separated. So here with these red chromosomes, one pair is being pulled to one pole and the other pair is being pulled to the other pole. Telophase is the opposite of prophase. So you're going to have the nucleus reform. The chromosomes are going to kind of dissolve back down into chromatin. The spindles are going to disappear. And this is where you also start seeing cytokinesis. This is where the cytoskeleton starts to get tight in one area, and then it pinches apart. And when it does, that pinching is going to create two separate cells, and they're called daughter cells. Another good example here. So this dotted line you see here, that's where the protein's getting tight, and then that's where the cell is going to eventually pinch apart. We looked at meiosis compared to mitosis because a lot of people get these two mixed up. So meiosis is the copying or the creation of an identical cell. Meiosis is the halving and creation of a sex cell. So very the two processes have similarities. Let's see. So this top one showing you meiosis, and this bottom one showing you mitosis. So they both have subphases that are classified the same: prophase, metaphase, anaphase, and telophase. With mitosis here, you can see it's just one phase and then one cytoplasmic division. With meiosis, it's two phases, and at the end of each phase, we have a cytoplasmic division. So that means the cells produced with meiosis is always four cells, and the cells produced with mitosis is always two cells. And then with the actual things are happening with the subphases, they're very similar. So prophase, you would expect nucleus to disappear, chromosomes to condense, spindles to be formed. Metaphase, they would line to the equator. Anaphase, they'd be separated. Telophase, they split apart with the cytokinesis. But some special things that are happening with meiosis that doesn't happen with mitosis is a crossover event. So crossover is happening up here. This only happens in the first phase of meiosis. So this is where you have the blue and the red. So we'll say the blue is the paternal chromosome from the father, and the red is the maternal chromosome from the mother. You can kind of see here that they swapped a segment of chromosomes. So this swapping or kind of shuffling up like a deck of cards is called prophase. It's called crossing over. And then the really difference between the anaphase and the anaphase 1 and anaphase 2 is anaphase 1 separates the pairs. So you have the big X's separate first. And then over here in anaphase 2, you have the chromatids, sisters separate. So that the individual separated chromosomes in anaphase 2. From meiosis, we can kind of move on to genetics. Close some of these folders. Let's see. So with your DNA, not every cell in your body uses the same DNA. Some cells, like the eye, for example, won't be using some of the genes that the liver would need. So they pick and choose what DNA to use. So this process is called differentiation. It happens very early, so as the zygote forms. So you can see here these first two are kind of a pinkish color, maybe tannish. These cells are called stem cells. They have not been activated just yet. And as they get a little older, you can kind of see the colors went from that tannish color, yellow, to blue, red, and green. So they started to differentiate. Blue is going to be things like skin, red is going to be things like muscles, bones, and the green is going to be things like your digestive tract. And then once they pick what they want to become, they can't go back and pick a new job, basically. They're stuck that way. So we know this based on some science that Mendel or Gregor Mendel did. Gregor Mendel is referred to as the father of genetics. His most famous experiments were done with pea plants. He went around and hand pollinized pea plants, and he looked at the offspring created. So pea plants have many different traits. Um, for example, he looked at the seed colors, so yellow and green here. So when he crossed a yellow and a green plant together, they were always coming out yellow. And then if you cross these individuals, it's called a back cross. The offspring produced from that back cross, you have the yellow, and then some of the green reappeared. So this showed him that there's something coding for those colors, and that the code sometimes can outweigh or overshadow one of the genes. So in this case, the yellow is going to be referred to as a dominant gene because dominant is going to overpower a recessive, and the green is my recessive this case. If it does get paired with a dominant, it's always going to be overshadowed by the dominant allele. Dominant genes are written as uppercase letters, and then lowercase letters are going to be used for my recessive alleles. see, where's my phenotype stuff at? Oh, I'm blind. There it is. Um, so when you're referring to the code, the A's, the big A and the little a, that's going to be called a genotype. And the genotype is going to like be a written down code that you can use for reference. A phenotype is the physical trait that's going to be used or produced by the organism. So if I had a over here, I have YY and YY. So the yellow in this case is the dominant large case Y. So that's going to be yellow. And then two small case Y's or lowercase, that's going to be green. Homozygous is where you have two individuals of the same letter. So over here with this two small Y's, two uppercase Y's, these are homozygous. Down here where you have the uppercase and lowercase Y, that's going to be heterozygous. You can test probabilities of offsprings if you know the parents' genotypes with the Punnett square. I think we've done enough of those. There's uh, not all genes are going to follow Mendelian genetics. There are some special ones out there. So one case is where you have two dominant alleles, and if an individual gets both alleles, they're not going to both be shared. Like for example, if you had a white and a red flower, you don't get splotches of white and splotches of red on the flower. They actually blend together. This is called incomplete dominance. In this case, the red and the white make a pink flower. Then you have the ones that do not blend. So in this case, you would have patches. So we have a good example is cow fur. So if there's a red cow and a white cow, um, codominance, the cow is going to come out with red patches and white patches of fur. Similar to that with our blood cells, um, if somebody's born type AB blood, that means they have a section of the blood cell. Some of it's going to have the A protein and some of it's going to have the B protein. So it has spotches of A and B. Okay, transcription translation. This is how you take DNA and make a protein. So DNA comes in different forms. The linear form where it's straightened out in this helix, this is called chromatin. But chromatin isn't a good way to store DNA, or not to store DNA, move DNA. If you were to move chromatin around, there's a really good chance you could rip or tear. So the best way to move DNA around is in its solid form, and the solid form condenses and wraps around itself till it becomes this solid structure down at the bottom, and the solid structure at the bottom is called a chromosome. Chromosomes are best for moving, but chromatin is best for reading and copying. So when you want to read it, you start out with the chromatin, you do a process called transcription, and then with transcription, with this arrow, you're going to get RNA. From RNA, you're going to do a process called translation, and from this arrow, you're going to get a protein. So with transcription, literally it means to change word, not change language, but change word. So both DNA and RNA are still written in the nucleic language. But translation means to change language. When you go from RNA to protein, you're changing the language from the nucleic acid language to the amino acid language. We, when you go through transcription, you don't transcribe your entire DNA. In fact, most of our DNA is often referred to as junk DNA. Only the gene segments are going to be transcribed to use for proteins. So you're going to read a little section of your DNA. This is going to be RNA polymerase. This is creating my messenger RNA. And then from this, the messenger RNA is going to meet up with a ribosome. So the big blue structure is my ribosome. The messenger RNA is down here inside of it. It's going to read that for instructions. You also have another RNA molecule called transfer RNA. This molecule is bringing in an amino acid. And these three together are going to build my protein, kind of like a 3D printer. And it's going to print my protein. It uses the three-letter codes on the messenger RNA as instructions to bring in an amino acid. So these three-letter codes are called codons. Every gene that we have has the same start codon. So this down here, the green AUG, it's always going to start a protein sequence, and then they all stop the same way. They have three options to stop the same way: UAA, UAG, and then the UGA. So all three of these are going to reference the stop of the protein to terminate the translation process. So we can manipulate the fact that we know how transcription translation works. Some things that we can do is if you want to copy DNA, right? Let's say that you have a very small segment of DNA, maybe from a crime scene or something, and you want to test it. If you were to test it, you might destroy it. So what you can do is copy it first. To copy it, it's called PCR, the polymerase chain reaction. It kind of works on synthesis of DNA. So you're going to heat up the DNA, and as you heat up, you're going to break up the segment. And then the two segments are apart, and as you cool it back down, they're going to want to join back together. But if you also throw in primers in the solution, there's a chance that the primer is going to bind to the DNA segment, and that's going to keep it from binding to the original. And now that these two are bind, you're also going to have polymerase in there. So polymerase is in there. It's going to read that there needs to be some nucleotides um, put in the segment here to complete it. So the DNA polymerase is going to join there, and it starts to read and fill in the gaps. So after one segment, you've doubled the DNA. So that's just one cycle. You do this for 20 to 30 cycles, maybe even 40. Each time you do, you double your DNA. So you're going to have billions of copies of DNA by the time you're through with PCR. We can also clone genes. We have these things called restriction enzymes. They cut out genes. So in this case, we took a foreign DNA. I think I used the bovine growth hormone as an example. So you can take out the bovine growth hormone from a cow, glue that into plasmids of bacteria, and then reinsert those plasmids back into the bacteria cell. And when you do, you're introducing a new gene into those cells. And when they go through transcription translation, they produce that bovine growth hormone, and then you can mass harvest it. It's called gene farming. Stem cell usage. So again, these cells in here are yet to differentiate. You can actually take stem cells, put them into an environment like the spinal cord or into joints, and the environment that they're in is actually going to stimulate them to become new cells. It's a good way to try to repair tissue that's very slow at repairing, like the spinal cord or joints. Gene therapy is a good way to introduce a gene into a cell that is in need of a gene. So if a cell or somebody's born with a defective gene, you can use a virus. So you have to take out the bad DNA that's going to harm the cell, and you swap that in for a good DNA. In this case, a working gene. And that virus is going to infect the cell, and when it infects the cell, it's going to infect it with a working gene. The problem here is most of the cells that get infected are not mitotic, so the cells don't have a base to go through reproduction and copy the gene over and over again. Meaning that most people that go through gene therapy, they have to be infected over and over again with the virus to get that gene inside their body. And then cloning is something that's often referred to as a successful biotechnology. So cloning is very simple in nature, but it's hard to do. You have to have a zygote. As soon as the zygote's created, you take out that zygote nucleus, and you're going to insert a donor nucleus. So over here is another sheep in this case. This nucleus is removed from the donor cell, placed into the zygote cell, and the zygote's going to start to grow. And as it grows, it's going to use the instructions from the host as differentiation, and that's going to pretty much form the exact same structure that the host created because it's using the same set of instructions. Okay, we moved on to evolution because evolution and DNA go together. Evolution is nothing but just small changes in DNA sequence. So if it is a change in DNA, that's actually called mutation. And you can see evolution and large scale with the buildup and buildup of mutations over time. So they do have to affect the the individual as a population. Um, Evolution is going to be driven by natural selection, meaning that the environment is going to pick and choose what traits are beneficial and what traits are not beneficial. If you are beneficial at the given time, you're going to have success and you're going to grow and you're going to be kind of outcompete the other individuals. If you're being selected against, that means you're going to die. And if you die, you're not able to pass on your genes, which means you're not going to have any offspring with those genes in the population anymore. As the environment changes, in this case with the moths, the selection force might change as well. So these moths that are black were very successful in like a natural forest, but these white peppered moths were more successful when the forest was paved over and you had concrete buildings. Evolution was postulated by Darwin. Um, he went around the world on the Beagle collecting samples, and as he collected samples, um, he sent them back to Britain. He, um, what was supposed to be like a 2 year trip turned into a 5 year trip, so he had a lot of time to collect samples. When he got back to Britain, he actually sat on his information for a while. Not until this other man, Alfred Wallace, came along, who kind of helped him along with the idea. He and Darwin together both wrote both on the origin of species, and this is pretty much um, the whole theory behind evolution. Darwin's most famous stop was the Galapagos because of those islands. These are just some evidences for evolution. You have homological structures, so organisms that are related to each other should have similar structures, like limb bones of mammals. Same thing with homologies here. You can look at the development of these individuals. These are all vertebrates. They all develop the same way at the very start. So it's kind of showing you they all start out the same way. The fossil record is another good example. You can see how organisms have changed over time based on their fossils. We can use this information to define species. So a species is going to be the most specific level of taxonomy. With your species, you should only have one individual that is consistent of that species. Uh, Most individuals are going to use the biological species concept to define a species. And this is just stating that if two species can kind of breed together and then when they produce an offspring, that offspring is fertile, that means they're the same species. For example, there are some individuals that can breed together, like a lion and a tiger, a horse and a donkey. They can all breed, but their offspring, the liger and the mule, they are infertile. So they don't have the ability to produce any offspring because their chromosomes don't exactly match up. And this is taxonomy going over the trees. So these are the levels of taxonomy going from broadest to most specific. When you're reading a tree like this, the closer individuals are on the tree, like the rabbit and lizard here, the more closer they are related to each other. The farther you go back down the tree, so if you had to compare the rabbit to the rabbit and the fish aren't closely related to each other because they're way down the tree. In this last one, we talked about bacteria, protists. So again, bacteria should be prokaryotes. We have two general types of or prokaryotes, bacteria and archaea. So archaea are those special ones that are referred to as ancient or extremophiles. They can live in very rough environments like volcanic beds, hot geysers, poison methane swamps. Uh, if somebody does get sick with bacteria, bacteria can a lot of the time be treated with antibiotics. So antibiotics themselves do not kill bacteria, but they weaken bacteria. They slow down their reproduction. They slow down their biosynthesis. It makes it easier for your body to remove the bacteria. The bad thing about antibiotics is they do mutate very quickly or they become resistant very quickly because of the way bacteria grows. And then antibiotics can also kill the good bacteria, the probiotics in your body. So there's always a kind of a downside to that. We talked about the protists or the protistas. So all these individuals here are going to be microscopic eukaryotes. So each one of these guys have a nucleus. And then we moved on to fungus. Again, fungus are also going to be eukaryotes. These are more macroscopic. When it comes to a fungus, you're going to have them having hypha, which are kind of like roots. So again, these are not plants, but they have something very similar to a root. And then the collection of hypha is the actual fungus, and that's the mycelium. So several phyla here of the fungus. Most ones people recognize are going to be this last one here, the Basidiomycotas. So these are going to be the mushrooms that everybody sees. So mushrooms are going to be the reproductive part of the fungus. These are going to be the spore producers. So if you look underneath this cap of the mushroom, you're going to see gills. And if you were to tap it or kind of give it a pat, it'll look like dust coming out of the mushroom, but those are actually spores, and the spores are used to reproduce with. Okay, we're kind of running out of time here, but I kind of get most of that covered. Anybody have a question?
Prof,
What's up? So I did create like a little quiz for you. What's the best way to give them that quiz, Harry? Just you're muted.
Is this the whole material for the Gen Bio at the college level? Ken?
It's sort of college level, yes. I kind of reworded it to make it maybe a little easier since they're not um in college just yet.
So
It did
it
have
will be Bio 1110?
um yeah, 1110. This is all Gen 1 stuff.
110 at the college?
College level, yep. I mean, I can send
Okay.
it to you through the WhatsApp app, and then you can try to give it to them.
Mhm.
Okay.
So what is the plan next? Next week will be test or exam?
Yeah, we can do it. I can. Blah, blah, blah. You want to just have them working on the entire week? They still testing all next week?
There'll be, or, okay, if you plan to meet again next week, is that right?
Well, we don't have to meet. No, we can just do the test. Just
Okay,
normally,
test, yes.
yeah. Normally when we have a test, we don't meet. We just let them do the test.
Right. So let's do the test next week.
Okay, that'd be fine.
All right. Okay, guys, you try to do the best, okay? I was wondering if they have the review material, the
Um,
chapters.
well, they have access to the book, the OpenStax book.
Okay.
Everything I pulled from my review is in that book.
Right. So Alfonso, you have the book, right? Honey, you have the book.
udah.
Yeah, the ebook. Okay, that's good. That's good. So hopefully,
Terima kontak.
okay, guys, this is a college best that you can. So you are like 7 years, okay, ahead, right, Ken?
Yeah, they're big. They're pretty much jumping ahead here. Yes.
Right. Okay. 7 years ahead. Okay, we need to keep it up because you guys are special.
Yes.
Just remember. Okay, remember that. All right. I think, do you have any other things, Ken, to share or?
No, I'm good. I'm gonna jump off.
Okay.
All right,
Thank
thanks again,
you.
Harry. You're welcome.
Ya.
Okay, everybody say bye. Okay.
Iya.
Yeah. All right. 老师 here. 老师你好。
Hello，你好，Sir，大家好。
Dia bukan.
Bánh kem. So, Lause kapan mereka akan ujian Lause?
Untuk yang HSK 0, betul, sir?
Iya, HSK
Yes,
0.
I'm leaving now.
Ya, HSK 0. Ini Ananda udah di sini. Ya. Dan juga kita minta ada yang grup Mandarin, bahasa Mandarin yang buat orang dewasa. Orang dewasa, kita juga udah minta mereka untuk ikut ini. Lause Jen yang ngajarin.
Oh,
Ini pengucapan, pelafalan. Like, gimana ngomong dengan menggunakan napas dan sebagainya. Oke, Ananda. Mana Ananda? Nah, ini sama
Oh,
Ananda. Oke, Ananda ini ikut Lause Kensi, ikut di sini. 2 ya, biar cepet. Oke.
ya, sir.
Mhm. Ini kelas berapa Ananda? Kelas berapa?
Kelas 2.
Kelas 2 ya? Wah.
Udah naik kelas.
Mhm. Sebentar lagi udah naik kelas.
Bentar lagi udah naik kelas 3.
Iya. Nah, Ananda di sekolahnya namanya apa?
Mandarin tadi, belajar Mandarin di sekolah. Pernah belajar
After.
Mandarin.
Apa nama sekolahnya?
IPK itu dekat
Hmm,
dekat
IPK Bekasi ya?
itu, yang di Bekasi itu.
Iya, 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 IPK. IPK kan sekolah bagus ya, Lause.
ya.
Salah satu yang bagus lah. Ya, jadi, oke. Ini ya, lah, tadi lupa di off dulu. Ini, oke, di stop recording dulu ya.